Hi, and welcome to our lecture on bone tissue and structure. We're going to be taking a look at the kind of microscopic structure of bone in this, as well as some features that are more macroscopic so we can understand what exactly a bone is. We're going to begin here with some skeletal functions, and these 5 functions should be pretty darn easy to understand. Number 1, of course, we think about our skeleton providing us with support. So imagine not having bones inside of yourself and how you'd be kind of this, bag of muscle and goo. And that would make things a little bit difficult, including movement, which is our second function. If we think about how muscles work, muscles like our biceps brachii here can be attached to skeletal landmarks on one end and another skeletal landmark on another end with a joint in between such that when the muscle contracts, it's going to take this set of bones and bring them closer to that other attachment end. So when we learn about muscles, we'll learn about what we call origins, the stable end at which they attach, and insertions, the moving end where they attach, and how the muscle contraction tends to move those closer together. Now, along with support, these rigid structures that we call bone can provide protection. And our skull, of course, is a great example of that. But our rib cage as well as our pelvis also can protect some of the organs within them. And then we get to the things that are kind of a little bit different. It ends up that bone is, of course, a mineral storage area. If we're going to put things like calcium into our bones, that means that we can put calcium in and we can take calcium out. And we do need calcium for other purposes. Calcium is essential for things like muscle contraction. So if our blood calcium levels are going to drop, we can actually tap into our bones to get more blood calcium. And when we consume calcium in our food, we can redeposit that back into the bones. We also store in the yellow bone marrow some fat. So our bones can actually store fat inside of them. Even bones like our radius and ulna of our antebrachium, which seem relatively small, if you slice into those, you're going to find fat inside of the hollow spaces. And finally, our bones are involved in what we call hematopoiesis. This is blood cell formation, and it's important to recognize this is producing both red blood cells, and obviously the red bone marrow might make us think about red blood cells, but it's also about producing all of our blood cells. That means that we're producing white blood cells, we're producing platelets, all from common stem cells that reside in our red bone marrow. So what exactly is this bone stuff? Well, it ends up that bone is composed primarily of 2 substances, inorganic and organic components. And we kind of would expect this because we know that bone is a connective tissue. We're going to have a ground substance, and that is made of hydroxyapatite. And this is about kind of 2/3 of the bone. It's a combination of calcium phosphate and calcium carbonate to give us that kind of crystalline structure of bone. That's the ground substance. And then, of course, connective tissues have some kind of protein fibers. And we have large collagen fibers forming about 1/3 of the bone, which are going to provide a little bit of strength to that, because obviously a crystalline structure would be rigid, but it would also be brittle and easy to fracture. And the collagen fibers actually make bone a lot more tough. It's basically reinforcing that rigid ground substance to make it a little bit more pliable or more able to withstand abuse. So we do have a number of cells associated with bone. One of our obvious questions on an exam, of course, would be, which bone cell does blank? And we're going to start with osteoprogenitor cells. These are ones that are progenitors, that they are going to produce new cells. So they divide to produce daughter cells that become the matrix producing cells, which we're going to recognize next as osteoblasts. So we locate these in the periosteum and endosteum. We haven't gotten to those yet, but we will soon. The osteoblasts lay down the organic component of bone matrix, and then that becomes calcified. So let's take a look at these 2 cells here. Here we have an osteoprogenitor cell. We are in the medullary cavity, the hollow space inside of a bone. So this layer of cells here is the endosteum, endo meaning on the inside. And in this case, the osteoprogenitor can divide to produce cells like these. These are osteoblasts. They are going to lay down this osteoid, this organic component of the bone, which will then mineralize using the calcium and phosphorus that are floating around in our blood. The third type of cells are going to be kind of different. They are the mature cells that sit in a lacuna, a hole within the matrix, and these are the osteocytes, and we see one of those down here. So in this case, this is isolated within a lacuna. It does have the ability to reach part of itself out through these what we call canaliculi, or little canals, so that it can connect to other osteocytes. An osteocyte has kind of the simplest meaning. It just means osteobone and cyte cell. So the blasts are going to be germinative, giving rise to new matrix. The cytes are then resident in there. They are actually the former osteoblasts that have been isolated within a lacuna. So let's take a look real quick here at the periosteum and endosteum. These are what we might consider to be membranes, although that is really only very true for the periosteum. It's going to be a tissue layer surrounding individual bones. You get the idea here that we can kind of peel this off of the bone itself, and that is absolutely true that you can do that. We can see that down in here. They've kind of cut a hole through there and are trying to get into the actual bone, but it's going to be continuous with joint capsules between bones. So we're going to see this fibrous bag on the outside, and it will be relatively thin in most places. It will thicken up in places like the joint capsules. The endosteum is a tissue layer where we house those osteoprogenitor cells. It's going to line the medullary cavity, the insides of these smaller canals, the central canals and perforating canals, but it's not this kind of fibrous layer like the periosteum. Rather, it's a single-celled layer, and it may even have some gaps within that cellular layer. So it's kind of a very different thing than the periosteum. But the two names are just letting us know that the endosteum's on the inside. Periosteum literally means around the bone. We do also have a fourth cell type, and this one has kind of been separated from the others because it is dramatically different. It's not different like I divide to give rise to stuff, so I'm a stem cell, I produce bone, I reside within the bone like our other three. This one is actually derived from the same stem cells that give us white blood cells, and it is essentially going to be a white blood cell that works within the bone to break down bone matrix. So it's kind of doing the kind of jobs that a white blood cell could do. Its job is actually really interesting. It can secrete acids to dissolve the bone, freeing up the minerals, and then phagocytize the organic component of that bony matrix. So the osteoclast here is a large cell in comparison to the surrounding cells, and you can see here that it has multiple extensions here so that it can be good at absorption. And the question is, why would this be important? And that, of course, is because we need to have a balance between osteoclast and osteoblast activity based on what exactly it is that we need. So homeostasis means the maintenance of a constant internal environment. In this case, we're talking about a normal blood calcium level. Calcium is Ca2+. That's the ion that roams around. If our blood calcium level drops, we're going to need to have our parathyroid release parathyroid hormone. Parathyroid hormone is going to cause a host of things to happen. It's going to cause the intestines to absorb more calcium. The kidneys are going to try to reabsorb more calcium so we don't pee it out. And this is going to kind of help us deal with kind of adjusting calcium levels, but those may not be quick enough because maybe we're not eating calcium at that time and those aren't going to be effective. But we can cause our bones to release calcium into the blood, and how do we do that? We stimulate osteoclasts. So that's going to cause blood calcium levels to rise. Now, of course, if they get too high, there's going to be kind of another side to this. If blood calcium levels are too high, we're going to stop absorbing it in our intestines. We're going to go ahead and reabsorb it less, and we can actually urinate some of that out, but we can deposit it into our bones for later usage by stimulating osteoblasts. So there is that balance between osteoclast and osteoblast. Now let's take a look at the structure of compact bone. So we've previously in our tissues chapter taken a look at cortical or compact bone versus cancellous or spongy bone. And compact bone obviously is based on a structure called an osteon. And we can see that in a lot of different ways. Here's a model from lab. We see a central canal, and here we see the concentric lamellae that surround it, and that tubular structure is our osteon. We can see one here in a scanning electron micrograph, and here under a light microscope. So it ends up that this cortical or compact bone is kind of one of the easiest tissues to identify regardless of what mechanism you're using to look at it. Now if we take a look at the osteon, we can again want to kind of identify those key components to it. We do have a central or Haversian canal, and this is going to be where we're going to have blood vessels and nerves traveling through. We're actually going to build this osteon around a blood vessel, and so that central canal is kind of very central, not just to location, but to the formation of the osteon. We have surrounding this concentric lamellae. These are layers, and concentric means that they are kind of circle within circle, or we could say nested circles. So close to the central canal, I have a very small layer, and I have a bigger layer around that, and an even bigger layer around that. Of course, I can see the lacunae where we have our osteocytes residing as well. So a couple of other layers we want to know about. In addition to the concentric lamellae that are part of the osteon, we have interstitial lamellae and circumferential lamellae. So the circumferential lamellae are these ones here towards the outside. In this particular model, we can see the periosteum here. And the periosteum is held in place by what we call perforating fibers. Those are tapping into the circumferential lamellae. Lamella, as it's written there, is the singular version of that. If you pick up a bone, the outside of it that you are touching are circumferential lamellae. Now, of course, we see osteons throughout the majority of this bone, but because these are circular structures, they don't necessarily tightly pack against each other. And the interstitial lamellae, meaning the between space layers, are the ones that grow in the spaces between osteons. So we can see those relatively clearly. Now, we also want to recognize the presence of canaliculi, or little canals, and this model happens to be a little bit better at that. We can see a central canal, and we can see arterial blood, venous blood, lymphatic vessel, and nerve all within that central canal. We have our concentric lamellae. We have various lacunae with osteocytes in them. And the osteocytes, you see in red here, this extension of plasma membrane where they can reach through these little canaliculi and maybe connect to each other, ultimately with some of them connecting to the central canal. Therefore, if we think about blood vessels being in the central canal, that's where the nutrients come from. Canaliculi allow all of these cells to ultimately communicate with that, even though that does mean they have to connect to each other. Now, we do have another type of canal, a perforating or Volkmann's canal. This is kind of the perpendicular version to the central canal. So the central canal runs through the tube of the osteon, but the perforating or Volkmann's canals are going to connect those central canals and ultimately, through what we call a nutrient foramen, connect to the outside of the bone. And this is a really important point to make. So here we can see a nice perforating canal. Here's a perforating canal, and the ones running up and down are the central canals. Over here we can see a nutrient artery and vein. We're going to have others up in here, and we would find a hole in the outside of the bone, a nutrient foramen, foramina being the plural of that, where blood vessels can go in and out. Why does this matter? Well, this is where we store our calcium. We need to go in there to pick that up or to deliver it, depending on what exactly is going on in our body. We, of course, have in these epiphyses, particularly the proximal epiphyses, red bone marrow, where we're forming new blood cells, and they need a way to get out of there. So it makes sense that we have to connect our bones to the rest of our cardiovascular system, and we need vessels to go in and out of them. So some general terms for long bones. We do have epiphyses, which are the ends of these, and we would of course recognize a proximal epiphysis closer to the origin. So when we take a look at a bone like this femur here, this head of the femur hooks to the acetabulum of our os coxae or hip bone, that would be the proximal end. And we're going to expect to find red bone marrow within the cancellous or spongy bone that fills this epiphysis. And you can see all of that cancellous bone in here with those little gaps in between the trabeculae. So trabeculae are the struts or projections that we see that form that cancellous bone and give it the mesh-like appearance that we see from it. Of course, this not only makes the bone lighter, but it also creates those spaces for that red bone marrow to exist in. Now, the opposite, of course, of epiphyses are the diaphysis. That's the central tubular portion of our bone, most of which is going to be hollow, forming what we call the medullary cavity, which literally means the middle cavity. So this is, again, that hollow space inside the bone. Kind of note that word medullary. We're going to use alternate forms of that, medulla, to describe the middle layer of organs later on, such as the renal medulla, the cerebral medulla, or the adrenal medulla. And inside of this, we can find yellow bone marrow. And yellow bone marrow is kind of gross here. You can see this inside of a cow femur. It looks like somebody's packed some butter inside that medullary cavity. And you can see that they've scooped it out to show that medullary cavity. This is not butter, but yeah, yellow bone marrow is adipose tissue. So if you think about using a bone to make soup stock, the reason for that is this bone should contain this yellow bone marrow inside of it, and you're going to be putting fat inside of your soup. All right, so bone forms in a couple of different ways, and we're briefly going to cover these. Intramembranous ossification and then endochondral ossification. So we want to think first, what exactly is this name trying to tell us? Intra means within. And membranous, obviously referring to membranes. Ossification just means bone formation. We'll use that in both cases. So this bone formation process causes bones to form from mesenchyme, which is an undifferentiated, you might think of it as a stem or embryonic connective tissue. And this will be able to become different kinds of connective tissues. In this case, we have these mesenchymal cells, so we can think of it as primitive connective tissue. We have an ossification center within this where we start having our osteoblasts secrete osteoid, and then again that will start to eventually calcify. Now what ultimately happens as we start to do this and this spreads out is that we end up with membrane on either side with kind of bone in the middle, so it's like a sandwich. And we'll kind of see how that results in something particularly interesting as we do this later on. Endochondral ossification, on the other hand, says we're going to form this bone not within membranes, and these membranes are going to be connective tissue membranes, ultimately our periosteum, but in this case it's endochondral inside of cartilage. So most of our bones, the long bones, are vertebrae, but most bones in our body, other than say flat bones primarily of our skull, formed by this process of endochondral ossification. And we begin with a cartilaginous model. So we form a cartilaginous model of our bone. We start bone formation in the diaphysis, forming a bone collar on the outside, then a primary ossification center where we can begin to produce that medullary cavity. And importantly here, we're forming bone in the diaphysis, but not the epiphyses yet, and we start secondary ossification centers in the epiphyses. What is so essential about this is as we form those secondary ossification centers and start to form those epiphyses, which eventually fill with cancellous bone, we leave hyaline cartilage from that fetal model on our articular surfaces, and that becomes that smooth articular cartilage. But we also leave epiphyseal cartilage, or our epiphyseal growth plate. So we leave an area on either end of the bone where this can produce new cartilage, causing the bone to grow in length, and we can of course convert some of that new cartilage into bone, and our bone will get longer. And eventually we'll reach maturity and we'll convert that whole epiphyseal plate into bone and no longer be able to grow our bones any longer. So this epiphyseal plate is just that cartilaginous growth zone, and we can actually see where that was in a mature bone here and where that is in the bone of a juvenile. You can see how this head and this greater trochanter of this femur aren't really connected to the rest of this bone. You can wiggle those things around because there was a slab of cartilage in here where bone growth could take place. So epiphyseal plate exists in juveniles as that cartilaginous plate of growth, and it converts into an epiphyseal line in an adult where no more growth is going to be able to occur. All right, so we'll think about bone growth then, and of course bones grow in a couple of different ways. They grow in length from the epiphyseal plate, and we can kind of have an idea of how this works. We've got matrix production occurring here, so we undergo mitosis, we grow, we produce new cells, and we have this whole new slab of cartilage which is eventually calcified to create kind of an additional length of our bone. But we also grow our bones in width as we grow, and we refer to that as appositional growth. It grows the diameter of a bone in a pretty simple manner. So what's going to happen is we're going to basically be producing new bone, and we're going to grow that along the outside here to make this bone larger in diameter. At the same time, we reabsorb the inside of this using our osteoclasts to make that medullary cavity larger in diameter. What this does is allows us to grow our bone on the outside and make it bigger in diameter overall without having to dramatically increase the weight so that our bones get too heavy. All right, so what are our bones? Well, how do we classify them? How do we look at different kinds of bones? Well, we have some basic categories. Long bones, like the humerus we see here, or the femur down in our lower limb, or the radius and ulna of our antebrachium. Those are obviously long bones because they are long. They're the ones we've been talking about with distinct epiphyses on the ends and a diaphysis or shaft down the center. We also have short bones, and short bones really aren't going to be like that. They're going to be these little box-like bones like we have in our carpals, or if we were to look down in our ankle region in our tarsals. So they are definitely relatively cuboid. In fact, we have a cuboid bone in the bones of our foot. And these are going to be basically that nice cortical bone on the outside, and the entire inside of this will be filled with that cancellous bone. But it ends up when you start to have categories like long bone and short bone, you look at some other bones and you're like, man, these are weird. They don't really fit. So we have a category called irregular bones, which allows us to fit in other bones. And irregular bones are things like our vertebrae. This body of the vertebra is very much constructed like a short bone, but it has all these crazy projections on it for attachment to other vertebrae or attachment for muscles. So that's definitely weird. Sesamoid bones potentially are the weirdest of all. We have here our patella, and then you may have a different number of sesamoid bones than other people around you, particularly if you happen to be older. These are bones that form within tendons. So you can imagine we are having a tendon coming from our quadriceps femoris muscle group down our thigh, and it's going to hook to the tibial tuberosity, that bump below your knee that's on your tibia. And you are born without a patella, but it starts to form as those legs start to move. We also have what we call flat bones, which are great and easy to understand. Flat bones are the bones of our skull. We can fit something like our scapula into that category, or our hip bones, which have these broad, flat surfaces, often for muscle attachment. These also tend to be some of the bones that we are more likely to have red bone marrow in, producing red blood cells and white blood cells. And it's constructed of something we refer to as diploe as a result of that intramembranous ossification. So if we take a look at some of this, you'll see a sandwich. Compact bone on the outside with periosteum on the outside of that. Compact bone here, this is going to be, say, the inside of our skull with periosteum on the outside of that, which would be actually facing towards our brain in this case. And then we have this spongy or cancellous bone as kind of the filling to the sandwich. So this is our diploe. It basically means that rather than having the bone be, say, a tube like this, because it is a flat surface, it does have to still be hard on both sides, but it's also still going to have that cancellous bone to make it more lightweight and to allow for blood cell production on the inside of it and it just has this distinctive kind of shape to it.